Now, as we're doing these more complex genetic analyses, we also need to keep in mind that there are still some very valuable ways that have been used for quite a long time to determine the role of different traits, different genes, how they interact. Reminder, all phenotypes are a combination of your genotype and your environment. And so we could mathematically represent that by an equation like this. Now, actually putting real numbers on these is very, very difficult, but people are beginning to do it. But for hundreds of years, people have been able to get at least general ideas about how much a trait was controlled by genetics or how much it was controlled by the environment. Now, the amount controlled by genetics we call heritability, right? Because you don't inherit your environment, you do inherit your genes from your parents. And so heritability is the amount of phenotype that you get from your genes. And again, for some traits, heritability is very high, near 100%, but for other traits, it's very, very low. Now, twin studies are one traditional way, and these are still used today, but this is one way that we can tease apart the role of genetics versus environment. Fraternal twins are like any other sibling. They are simply two eggs fertilized by two sperm, and so although those siblings share a womb, they are no genetically more closely genetically related than any other two siblings. However, monozygotic twins, also called identical twins, result from a single egg and a single sperm that later splits into two. And so those individuals are genetically identical. So if we track them throughout their life, any differences that we see in them are going to be based on the environment that they are exposed to. And so this is particularly valuable when we have two individuals that for some reason were separated at birth and raised in very, very different environments. And there are some surprising similarities between twins that are raised in different environments. So we start to get a little bit of an idea about what part of their characteristics are genetic and what part are caused by their environment. Now, we can also do kind of the reverse of this. So instead of just doing two individuals we know are genetically related, we can kind of take a big data approach. And we can look at an entire population with wide diverse genetics and then look to see how they interact with their environment. And so we can measure the strength of selection that is based on that phenotype. But this requires a lot of groundwork, maybe some phenotyping or genotyping to make sure that we understand the genetic diversity of this big, big population. But both of those have been used for many, many years and are still being used today. One very interesting study about kind of the big picture over a very, very long term is a study that's been done in corn or maize. Maize is a common term for it outside of the United States, but we call it corn. And 100 years ago, actually 120 years ago, they started this experiment, and it's still going on today. So they started with a single kernel of corn. They grew it up, and so all of those kernels are going to be genetically identical. And then at each generation, they did two different populations. They measured the oil content in the offspring, right? And they realized that these offspring are going to be mixed and matched. There's genetics, there's another parent contributing in there, but they all start out right at about the same amount of oil. And then they selected one population for increased oil and one population for decreased oil. And this is not, this is not a, an advanced or a very technologically complex study. Basically, all they had to do was have some way to measure the oil content and say, let's take the 10% of seeds from this crop with the highest oil and we'll plant them in one field. And then we'll take the 10% of the seeds with the lowest oil and we'll plant them in another field. And then we just continue that generation after generation. And so even after 100 years of selection, so this is the range of the beginning population, we have reduced it not quite to zero, but very, very low oil content. And we've raised it all the way up in the other range. But notice there's still diversity. Even down here with this such a low oil content, there's still some diversity. So even after 100 years, we have not yet fixed all of the alleles that maximize oil content. And so for these polygenic traits with maybe very complex genetic underlying architecture, it's actually going to be very, very tricky, maybe even impossible, at least in realistic scopes of time for if humans are doing the selecting. It's pretty much impossible to eliminate all of the genetic material. And there have been some species that we have been selecting for quite strictly for hundreds and hundreds of years, like domesticated crops, dogs, horses, cows, all of our domesticated animals we've been selected for, and in some breeds quite strongly for hundreds of years, yet there's still genetic diversity. Now we've got lower levels of genetic diversity than when we started with, but still genetic diversity. And so we realized that all of these complicating factors, right? Going back to Mendel, he was a genius and way ahead of his time, but he was just looking at the foundation, the beginning of genetics. There are all sorts of other things that play a role, some of which we've talked about and others we've just mentioned, and a few that we're really not going to get into. So I don't want you to spend time memorizing this list. Hopefully some of them look familiar, but just realize that it's incredibly complicated. And so if I want to say, oh, I want to figure out all the genetic contributions to Alzheimer's disease, that might be the work of many, many labs over, maybe even dozens of careers before we start to get a really good handle on that because of all of this complexity. Okay? But there are ways that we can at least begin to visualize it. And so we're going to look at an adaptive landscape. An adaptive landscape is a way to visualize, model, and maybe even as we gather really big data sets, do some quantitative work on populations. So in essence, what we do is we take two traits of interest and we plot them along the x-axis for one trait and the y-axis for another trait. And so every individual in the population we can represent with a red dot. So this is an individual with a short leaf and a short flower. Long leaf, long flower. Now notice these are correlated. If you have long leaves, it looks like you fairly have long flowers, but there's some spread and some diversity. Now, in the third axis, the z-axis, we are going to map fitness. So is it good to have short leaves and short flowers, or long leaves or long flowers? Which of these individuals do best? And so what we end up with is this three-dimensional graph, and this is called an adaptive landscape. Now, for some traits, this is supposed to be hidden at the beginning. So for some traits, it's very simple. There's only one fairly narrow range of traits that's the best. And if you get too far on either side, so if you have a long flower or a short flower, if you have a long leaf or a short leaf, anywhere, you're going to have low fitness. But if you have right in the middle, that's going to be good. And so this is probably a sign of very strong stabilizing selection, right? So the leaves and the flowers are adapted to do very well in the environment, and individuals with extremes just don't do very well. But for some traits, it's a much more complex adaptive landscape where we might have other combinations that are okay, and then one combination of traits that's overall the best. Now, natural selection tends to push populations so that they cluster around whatever is going to be the highest adaptive peak, the best combination of traits that an individual can have. And we might get some variety and some diversity there, but we're rarely going to get individuals way out here. Now realize that this adaptive landscape is going to be dependent on environment. If environment changes, then the combination of traits that is best, that is most adaptive, will no longer maybe be the best combination. And so environment can actually change this landscape. And if that occurs, then the population will remain where it is initially, but there may suddenly be a better combination of traits, a higher fitness peak. And so natural selection will work to move them up towards that higher fitness peak. However, sometimes because of this changing landscape, a population could get stuck on a local fitness maximum, some combination of traits that's pretty good, but not the best. And so to get to a new trait, we have to have one of two things. Mutation could take us there. We get a mutated individual with a very extreme phenotype that normally wouldn't be produced. And that might do it, and then he's going to reproduce with these individuals. Some of the babies might have that trait, and eventually we could then relocate the population to this combination of traits that is most fit. Genetic drift can do that somewhat also, but genetic drift needs to be very, very strong. So natural selection tends to push populations towards the top of whatever slope they're already on. Genetic drift allows for random spread, and sometimes that random spread is good. If the random spread is out here to a low point, those guys just won't do well and they'll die out. And genetic drift can do some spreading also. So all of those evolutionary forces have a different impact on the population. But by and large, especially given large periods of time, the population will cluster around some maximum with the diversity kind of spread around that, but not too extreme. And so this can happen over many, many hundreds of generations, or it can happen rapidly if we have one single mutation. And so making these large jumps is really kind of the key. Now, Darwin didn't really talk about this, but it's been revived several times, and there are actually some good indication that it is possible to make really large jumps, sometimes with a single mutation in a single gene. If it's a gene that is important enough, particularly one that is active in early development, then it might make a really, really large phenotypic shift. Now, these large phenotypic shifts are almost always going to have an impact on the fitness of the organism. And the vast majority of the time, they're bad, right? And so we see that all the time, right? In the human population, we see individuals born with what we call birth defects. A birth defect is simply a detrimental mutation. And so if you're born without eyes and you're a rabbit and you need your eyes for sight, then that's a bad thing. And the vast majority of these large jumps are bad. You're just going to kind of randomly land in some part of the adaptive landscape. But every once in a while, a little tiny genetic change makes a big phenotypic change, and it results in a big, big advantage. And so whether we have a very simple landscape or a very complex landscape, we can make these movements because of mutation. Now, diversity in populations like this are going to be high, particularly if these peaks are rather close to one another. Then portions of the population may be adapted to one peak, portions may be the other peak, so we might get a range of different phenotype combinations. And so if we have a simple adaptive landscape, we'll have a population with relatively low genetic diversity. But if we have a very rugged, complex adaptive landscape like this, then we'll have lots and lots of diversity throughout the population. And so these are going to result in populations that look quite different from one another.